In the complex world of modern pharmaceutical manufacturing, the pursuit of absolute 100% chemical perfection is a thermodynamic impossibility. What we call purity in a laboratory is never a pristine natural state. Instead, it is a rigorously engineered threshold, an acceptable limit of imperfection. To understand this threshold, we first have to separate the actual drug, the active pharmaceutical ingredient, or API, from the purposeful excipients, like binders and fillers, used to construct the final formulation. Once we isolate the active drug and the intentional additives, anything left over is an impurity. These are unwanted foreign chemical substances that remain inside the API or final product after synthesis is complete, or even develop during storage. These contaminants carry serious operational risks. First, they can trigger chemical instability within the mixture, drastically reducing the drug's effective shelf life. Second, they can degrade the drug's overall potency. If an active ingredient breaks down by interacting with a contaminant, the intended therapeutic effect is lost. But the most critical risk is biological. Certain contaminants are inherently toxic, meaning their presence in the final dose directly threatens patient safety. Because contamination is both inevitable and dangerous, pharmaceutical manufacturers build a specialized analytical matrix to actively identify, quantify, and strictly control these microscopic threats. This analytical matrix is a structured system designed to classify every possible contaminant based on its specific chemical behavior. This technical chart categorizes the primary chemical classes of these contaminants. We start with the most common classification, organic impurities, which are defined as carbon-containing compounds. Organic impurities are highly prevalent because they are often structurally similar to the active pharmaceutical ingredient itself. They typically manifest in two ways, either as unreacted starting materials left over from the synthesis, or as degradation products that form as the drug breaks down over time. Moving past carbon structures, the second major classification is inorganic impurities. These are strictly non-carbon compounds. In physical terms, these manifest as residual heavy metals, salts, and microscopic filter aids that enter the chemical mixture. Unlike organics, which are tied to the drug's structure, inorganic impurities usually stem directly from the reagents or metallic catalysts used to drive the initial chemical reaction. The atomic structure of the impurity, whether built from carbon or metallic elements, dictates exactly how a technician must hunt it down and isolate it. The third classification covers residual solvents. These are volatile organic chemicals used strictly to facilitate the manufacturing process. Solvents are intended to be completely evaporated out of the final product, but trace amounts stubbornly remain in the bulk material. Looking at the classification chart, regulatory bodies enforce a strict toxicity tiering system for these solvents. Class 1 solvents, like benzene, must be strictly avoided due to their severe toxicity and known carcinogenicity. Contrast that with Class 3 solvents, like ethanol and acetone. These have lower toxic potential, but they still require strict quantitative testing limits to ensure safety. Next, we have elemental impurities. Heavy trace metals like lead, mercury, cadmium, and arsenic pose a severe systemic threat because they accumulate in the human nervous system. We also account for physical impurities. These are macro-level foreign contaminants introduced from the environment, including ambient dust, glass fragments, or microscopic clothing fibers. Finally, microbial impurities present the threat of biological contamination from bacteria, fungi, or endotoxins replicating within the formulation. Classifying these varied contaminants assigns a specific severity tier to the biological risk each one poses to a human patient. Contaminants do not spontaneously generate in a vacuum. They exploit specific vectors of infiltration across the pharmaceutical supply chain. The first vector is the raw materials. If the initial starting substances arrive at a facility already contaminated, the entire downstream product is compromised before synthesis even begins. The second vector is the manufacturing process itself. Incomplete chemical reactions or unexpected side reactions inside the reactor will consistently spawn unpredicted compounds. The third vector involves storage conditions. Even a perfectly formulated drug can degrade into toxic impurities if exposed to excessive heat, direct light, or ambient humidity. The fourth vector is packaging. Over time, microchemicals can slowly leach out of plastic containers or rubber stoppers directly into the liquid drug solution. There is also the distinct risk of cross-contamination, where microscopic particulates from an entirely different drug batch are accidentally mixed into the current production run. And we must account for human error, where poor manufacturing practices allow physical environmental hazards to accidentally adulterate the batch. Because contamination vectors exist at every stage of production, quality control requires a continuous, unbroken chain of analytical surveillance. To maintain that surveillance, manufacturers match the physical properties of the impurities to specific, high-precision analytical equipment designed to hunt them down. High-performance liquid chromatography, or HPLC, is the standard for isolating organic impurities. It excels at separating structurally similar compounds from the main active ingredient. Gas chromatography relies on a thermal vaporization process, making it uniquely suited to detect and measure the volatile traces of residual solvents. For trace elemental and inorganic impurities, laboratories use inductively coupled plasma mass spectrometry. This machine vaporizes the sample to detect heavy metals down to parts per billion. Meanwhile, UV visible spectroscopy is utilized to identify degradation products, based specifically on how those molecules absorb light. This analytical detection operates under a rigid legal framework. The International Council for Harmonization, ICH, provides guidelines that act as the governing law over analytical testing. These frameworks are highly specific. ICH Q3A governs new drug impurities, Q3C polices residual solvents, and Q3D strictly caps elemental impurities. The ICH guidelines are what convert raw, abstract laboratory data into legally enforceable safety thresholds. We can apply this entire matrix directly to a ubiquitous, real-world example, the synthesis of paracetamol API. During synthesis, the primary organic threat to this pipeline is a compound known as p-aminophenol. This compound is highly dangerous. It is a toxic degradation product known to cause severe nephrotoxicity, leading to direct kidney damage. To combat this, the manufacturing facility must deploy strict HPLC monitoring, ensuring the p-aminophenol concentration stays safely below its legal limit. The secondary threat to the paracetamol pipeline is acetic acid. Acetic acid is a residual solvent left over from the synthesis phase. Because it is volatile, it requires a completely different detection modality than the organic degradant. Technicians must physically route samples to a gas chromatography machine to vaporize and quantify any remaining traces of the acid. Releasing a single common API to the public requires running multiple, overlapping, and highly complex analytical methods simultaneously. The pursuit of zero impurities in pharmaceutical manufacturing will always be an illusion. True pharmaceutical quality assurance is the rigorous science of keeping inevitable contaminants quantified, controlled, and well below the threshold of biological harm. Executing the impurity matrix is the non-negotiable safeguard standing between complex chemical engineering and global patient safety.